We also have various chemicals within our first line of defense that can block pathogens from getting deeper into the body, one being the enzyme lysozyme. So the enzyme lysozyme is predominantly found in tears and saliva, but we do have a small concentration in sweat as well. But this enzyme is able to break down the peptidoglycan layer in the cell walls of bacteria. So when bacteria are subjected to lysozyme, they essentially burst through cell lysis as their cell wall becomes destroyed. We also have a low pH due to the acid content on our skin. So generally, our skin has a slightly acidic pH. Most bacteria prefer a more neutral pH type of environment to survive in. So if a pathogen would land on our skin, it's generally not going to be able to thrive because of that acidic pH. And then in the stomach, we have gastric juices, which have a very low pH, anywhere between 1 to 3, and that is destructive to, again, most bacteria. Most bacteria would not be able to survive in that low of a pH. There definitely are exceptions to the rule, or else we wouldn't have GI infections, but most microorganisms are not able to thrive in that type of an environment. But again, bacteria like Staph aureus, the bacteria that causes botulism, which is Clostridium botulinum, and even a bacteria known as Helicobacter pylori, which causes gastric ulcers. All those bacteria are unfortunately able to survive in acidic environments. They are considered acidophiles, so they would be able to survive there and cause GI infections. But other types of bacteria, especially those that prefer a more neutral pH range, they're not going to be able to survive in the high acid content of the stomach. So the second line of defense, this is going to be defenses that are more internal, and it's going to be different processes as far as cellular processes and the accumulation of fluids from things like inflammation that are going to block or protect us from microbial infection. So we want to think of these as more as a kind of deeper internal mechanisms to protect us. So unfortunately, if a microorganism is able to get through the first line of defense, like the skin or nasal hairs or whatever it be, this is when our second line of defenses will now start to kick in. And the second line of defense is going to be the phagocytic cells. So phagocytosis is a process whereby certain white blood cells called phagocytes engulf and destroy infectious microbes. So there are 3 different classes of phagocytic cells: neutrophils, monocytes, and macrophages. Neutrophils are definitely the most common phagocytic cell in our body. So this is the one that we have the most of, the highest amount of, and neutrophils are going to circulate through the blood and tissues in order to engulf and destroy pathogens. An interesting thing about neutrophils is typically when they engulf and destroy pathogens, they also die themselves, and that accumulation of dead neutrophil and dead bacteria is where we get pus. So anytime you see pus coming from a wound, it's a good and bad thing, I suppose. Bad because you know you have an active infection, but I guess at least you know the neutrophils are on site trying to get rid of the infection, and it's our hope that the neutrophils are able to overcome the infection at that point. But if not, that's when other processes start to become activated in our body, like inflammation, and then eventually maybe antibody production in the third line of defense. Monocytes are phagocytic cells that are found exclusively in the blood. So monocytes are going to circulate throughout our blood, and they are going to engulf and destroy any pathogens that they come in contact with. They have a very important job because if anything pathogenic gets into our blood, from there it really has access to all different places in our body. So they are going to find those microorganisms in the blood and then engulf and destroy them there. But they are exclusive to the blood. Now they can leave the blood. When they decide to leave the blood, which we often call diapedese, so when they diapedese out of a blood vessel and then enter a tissue, whether that's lung tissue, skin tissue, whatever, but when they leave the blood and enter a tissue, they get activated to become macrophages. So monocytes are monocytes when they're in the blood, but when they diapedese out of the blood and enter the tissue, at that point they become macrophages. So macrophages are exclusive to the tissue, and they get called different things depending on what type of tissue they're in. So for example, if they're in the lungs, we call them alveolar macrophages. But they're phagocytic cells that are found within specific tissues. Again, their job is to collectively engulf and destroy infectious microbes. And they can typically work much faster and go through multiple rounds of phagocytosis before death, as compared to something like neutrophils, which typically can only go through 1 or 2 rounds of phagocytosis before dying themselves. Right here we are looking at blood. So all these are red blood cells throughout here, but then we do have a neutrophil here that's in the blood, and this neutrophil is kind of honing in on the bacterium here, and then there's another bacterium here. So this neutrophil, its kind of job at this given moment is to attach and then engulf and destroy the bacteria present here in the blood. So one thing we might ask ourselves is, how do phagocytic cells know what to engulf and what not to engulf? Because we obviously want them going around and engulfing infectious microbes, that's their job, but we don't want them to go around and engulf any of our healthy cells. So they need to know what's clear to be destroyed and what don't we not want to destroy. And this is all receptor regulated. So basically, any type of foreign pathogen like a bacteria is going to have a special marker on it called a PAMP, or a pathogen associated molecular pattern. So every type of bacteria, virus, fungi, they all have these surface markers on them known as PAMPs. And that lets our body know that they are foreign and they need to be destroyed. Phagocytic cells have their own receptor. They have a receptor called a PRR, or a pattern recognition receptor. So how it typically works is we'll have a phagocytic cell. So this is a dendritic cell, which is a type of a phagocytic cell, it's a type of macrophage. But we have this phagocytic cell, and this phagocytic cell has its PRR, and it is going to use its PRR to bind to a PAMP on a foreign pathogen. And once the PRR on the phagocytic cell attaches to the PAMP on the foreign pathogen, that's going to induce the phagocytosis process. Phagocytic cells are not going to go around and engulf your healthy human cells, because your healthy human cells don't have that foreign marker on it. Your healthy human cells do not have PAMP markers on them, so they're not going to be recognized for destruction. Whereas anything like a bacteria, virus, fungi, they would all have PAMP markers on them, so the phagocytic cell would know to bind to, engulf, and destroy those. So this is basically the mechanism of phagocytosis. So how does a phagocytic cell kind of work from beginning to end? So this is definitely something where you would want to know the various stages and potentially even the order of the stages. Fortunately, the names of the stages really do kind of summarize what's happening during each individual stage. But to kind of step back from it, again, really what phagocytosis is are phagocytic cells engulfing and then ultimately destroying things like foreign microbes. So the first stage is chemotaxis. So in chemotaxis, what's going to happen are chemokines, which are these chemicals in our body, are going to get released, and they are going to attract phagocytic cells to the site of infection. So if I have a cut in my finger, cells around the infected tissue in my finger are going to release these chemicals called chemokines, and that's going to attract phagocytic cells like neutrophils and macrophages to the site of infection. Because at any one given time when you're not infected, those phagocytic cells are just kind of evenly circulating throughout your body looking for potential pathogens. But if you have an active infection, let's say in the finger, that is going to induce the release of chemokines, which is going to draw those phagocytic cells like a magnet to the site of infection. So now I'm going to have this rush of phagocytic cells coming up to my finger. So the second step is adhesion. In the adhesion stage, that is when the phagocytic cell is going to use its PRR to bind to the PAMP on the foreign pathogen. So that's the PRRs binding to the PAMPs, and that's how the phagocytic cell is going to bind to the foreign pathogen, such as the bacteria. Then the third stage is kind of a twofer. It's engulfment and phagosome formation. So basically what happens is the phagocytic cell is going to extend its cell membrane and engulf the entire bacteria. So that's the engulfment component of that. So now the bacteria is inside of the phagocytic cell. Now when a bacterium gets engulfed into a phagocytic cell, we don't want the bacteria just to have free range and start swimming all over the cytoplasm. You want to restrict it so it doesn't cause any internal damage. So once the phagocytic cell engulfs the bacterium, it wants to encase it inside of a vacuole called a phagosome. So a phagosome is just a membrane-bound vacuole that is going to temporarily store the bacteria while it awaits destruction. So it's really just kind of like a holding area. Step 4 is phagolysosome formation. So in phagolysosome formation, the phagosome, which is holding the bacteria, is going to fuse with a lysosome, which the lysosome has all the digestive granules that are going to eventually be used to break down or degrade the bacteria. But when the phagosome fuses with the lysosome, you get this kind of hybrid structure called a phagolysosome. Step 5 is the killing stage. So that is when the digestive enzymes are released and they are going to degrade and effectively kill the bacteria. And then the 6th stage is elimination. So anything that the phagocytic cell feels that it wouldn't kind of recycle or reuse, it would exocytose out of the cell. So some things it would be able to reuse. So for example, things like nucleotides, it could potentially reuse for its own replication purposes. But something like peptidoglycan, which human cells have no need for, that would obviously be exocytosed out of the cell. This little video is a good one to kind of go through and watch on your own, but it kind of shows what this process looks in real life, so you can actually watch, I believe it is a neutrophil kind of chasing after some Staphylococcus aureus, and even see it engulf that Staphylococcus aureus and internalize it into the cell. You obviously won't be able to see all the various stages, but you can kind of imagine as the phagocytic cell kind of works its way through that process.